Okay,
Certainly.
perfect. Sure. So, can I share my screen? Like, may I share it?
Yes, sir.
Okay. Hmm. So, yes, good evening, everyone. And yes, welcome to yet another session in biochemistry, where I will be now quickly revising the important previous year questions which have been asked in the last 10 to 15 years. So I will be focusing on the important concepts, the important topics, the important questions that are potent for the upcoming university exam in August. So these series have been initiated from July 1st, right? And we are revising all the important questions in a very crisp, concise, and like conceptual manner, right? So yeah, the ones who are going to appear for the university exam in the next month. So this will be like a piece of cake, guys, for you to like quickly revise all the potent questions along with me. So these are the questions that I will be covering up today. That is regarding the chemistry of the carbohydrates and yeah, some important concepts that I will be discussing up today, including these questions, right? So these are the ones which I will be covering up in today's live class. Now, otherwise, guys, you all know about the core features of this app, right? We have the live classes that are streamed on YouTube right now, right? If you are like, if you missed it, you can just go to the recorded classes that are being available on the app. Apart from that, guys, you have the notes which are curated in the form of Q&A. That is the question and the answer format, right? And that is something which is very much beneficial as you will having the question directly, which is like So, and yes, after each topic, guys, you will be having certain MCQs from basic to advanced level, right? Covering all the important previous year topics, the previous year questions, right? All the clinical based questions, and that will be helpful to, yeah, score good. Now, this is the basic subscription plan that is available on the screen, right? You can see on the screen. Apart from that, if you want some extra benefit or discounts, like you can just use my code Dr. Abhijeet 20, right? So let us start with the classification of the carbohydrates. That is the very first question that I will be discussing up today. So before that, coming on to the classification of carbohydrates, guys, let us quickly revise what are the carbohydrates. Since we like so let us just revise what are carbohydrates, guys, first. So I will be coming to the question. The classification would be very easy, guys, if we just come on to the definition right now over here. Now, when I say carbohydrates, guys, so carbohydrate means they are hydrates of carbon. They are hydrates of carbon, guys. They are hydrates of carbon, right? They are hydrates of carbon, or the definition that is given in the book. They are hydrates of carbon, right? According to the name, if we see, it is these are the hydrates of carbon, right? Now, on the other side, the definition. So the carbohydrates are aldehyde, guys. Aldehyde. You already know what is the functional group, right? This aldehyde is the functional group. That is the formula over here. It is CHO, or I can also write it in the form of like this, that C. Okay, I can write it in this form as well, right? CHO simply, or C double bond O, obviously, and this is H. So that is nothing but the aldehyde group, right? CHO, or keto derivative or keto derivative. Now, when I say keto group, guys, I think you already know that is C double bond O, right? C double bond O, that is the keto derivative of polyhydroxy alcohol, of polyhydroxy alcohol. Now, guys, now let us just try to simplify the definition, guys. Now, when I say polyhydroxy alcohol, when I say polyhydroxy alcohol, you already know, yes, it will be containing OH group, right? Hydroxy means the OH group, poly means many. So let us take an example of polyhydroxy alcohol. That is basically the simplest one. That is the glycerol. That is the glycerol. That is the glycerol, guys, right? Now, once you understand the formation, guys, the monosaccharides classification will be very easy. I'll be like telling the names and it will be like easier for you to understand. Now, when we draw the basic structure of the glycerol, it is polyhydroxy alcohol. Now, it is more than one hydroxyl group, right? What does it mean, polyhydroxy alcohol? More than one hydroxyl group. More than one hydroxyl group, right? More than one hydroxyl group. So we will have, okay, let us name the carbons 1, 2, and 3, and we will have this, yes, OH group, the hydroxyl group, it will be more than one. And you already know the carbon forms four bonds, right? The valency is 4, right? So yes, in the first place, just to complete the valency, we have to add 2 hydrogen, second, like only one, and third again, H2, right? So this is the structure of the glycerol or the polyhydroxy alcohol. Now, what does the definition says? They are aldehyde or keto derivative of polyhydroxy alcohol. Aldehyde or keto derivative of polyhydroxy alcohol. Now, what we will do, guys, over here, yes, first we will make an aldehyde part right over here, the aldehyde, right? Now, we are just gonna remove the yes, the water molecule over here. I'm okay, the hydrogen molecule over here. Yes, in the first part. So one hydrogen from here and one hydrogen from here, right? So obviously you will get an aldehyde group over here, right? So remember, guys, it will be like C. Okay, at the first carbon, guys, you can very well see it will be like only oxygen over here, C double bond oxygen, and one H, right? Next carbon, again, it is same COH, right? And it is H. And the third one, guys, it is COH, and this is H2. Now, this is one which is containing this aldehyde group. How this group is formed? We are removing one hydrogen molecule, right, from the first carbon, and see the aldehyde group is formed over here. So this is actually called as glyceraldehyde. Glyceraldehyde, right? So glyceraldehyde, right? Now remember, guys, aldehyde or derivative group, it is always present at the first carbon, guys. It is always present at the first carbon, right? Now coming on to the keto group, guys. Now coming on to the keto group. For the keto group, what we are doing in the second carbon, guys, in the second carbon, we are removing this hydrogen group, right? From the second carbon, we are removing this hydrogen group. So that is again the keto wala part. Now, what it will be forming? Yes, C. Okay, the first one will remain the same. Yes, that is COH, and that is H2. The second carbon, since the hydrogen molecule is removed, there will be double bond with the oxygen. And the third one again remains the same. So yes, the keto group, it is at which position, guys? The second position. And remember, guys, the name of this compound. Yes, the name of this compound. It's very easy, guys. Now, this structure, guys, it is the structure of the acetone. It is the structure of acetone with two hydroxy groups. The one which I have encircled in blue, it is the structure of the acetone with two hydroxy group. So I can simply say it is dihydroxy acetone. Dihydroxy acetone, guys. Dihydroxy acetone, right? Dihydroxy acetone. So that is something you have to remember, that carbohydrates are aldehyde. Yeah, the very simplest one which I formed, glyceraldehyde, or keto, that is the dihydroxy acetone group of these polyhydroxy alcohol. That is something which you have to remember. Guys, apart from that, two important Viva questions that you can remember over here. The simplest carbohydrate, guys. Yes, the simplest carbohydrate that is over here, it is, yes, glyceraldehyde plus glyceraldehyde and dihydroxy acetone. These are carbohydrates formed from this polyhydroxy alcohol, right? Now, the parent carbohydrate out of this glyceraldehyde, it is the parent carbohydrate, right? The parent carbohydrate. And remember, the parent alcohol over here, The parent alcohol, it is glycerol. It is glycerol, right? From glycerol only we are forming glyceraldehyde, and this yes, this dihydroxyacetone, that is the keto wala group, right? So that is the basic definition. Now coming on to the question, guys, what is the classification of this carbohydrate? So let us just try to draw our own flow chart, and then we'll come to what is mentioned in the notes, guys, right? So yes, let us just try to discuss the classification of carbohydrates. Classification of carbohydrates. Now, we are dividing this, yes, carbohydrates into like 4 parts over here, guys, right? The 4 parts over here. Yes, first, second, third, and fourth. The first one, guys, we have the monosaccharide. Monosaccharide. The second one, we have disaccharide, right? We have disaccharide. Third one, we have oligosaccharide. Oligosaccharide. And fourth one, we have polysaccharide. Polysaccharide, right? So the carbohydrates, they are divided into monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Now, when I say monosaccharide, guys, monosaccharides in very simple language, this monosaccharides means mono means one and saccharides means sugar. So this contains single sugar unit. It contains single sugar unit, right? Disaccharide basically contains, di means two. So we have two potential sugar units, two potential sugar units. Then the third one, oligosaccharides. So basically we have the few sugar units ranging between, yeah, 3 to 10, right? 3 to 10, right? 3 to 10 sugar units, right? And poly means many, so it has more than 10 sugar units, more than 10 sugar units. So that is the basic classification, guys. Depending upon the number of the sugar units, they are classified into monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Now, guys, monosaccharides. Okay, let us just try to see the classification which is given over here. Now, in the monosaccharides, guys. Okay, let us just try to discuss a little about the monosaccharides. Okay, a little about the monosaccharides over here. Now, okay, now in the monosaccharides, guys. Let us use another slide over here. I'm discussing monosaccharides over here only. Monosaccharides. Just try to see, guys. So depending upon the number of carbon atoms, depending upon the number of carbon atoms. Now, the minimum carbon atom that we can take it is 3, guys. Yes, that is 3, guys. I've already told you the 3 carbon atoms, right? So we are taking 3 carbon atoms, right? So yes, we have 3 carbon atoms. We have 4 carbon atoms. We have 5 carbon atoms. Yes. And we have 6 carbon atoms, right? So the name given to the 3 carbon atoms over here, we are going to write it as triosis. Tri means 3. 4 is tetrosis. Tetrosis. 5, it is pentosis. And 6, it is hexosis. Right? That is basically the simplest one that we are taking up on the screen over here. Depending upon the number of carbon atom, it can be 3, it can be 4, it can be 5, it can be 6. Now, according to the definition of the carbohydrates that we have discussed bit over here, we have already told you they are classified as aldoses or they are classified as ketosis basically the two types of carbon atoms that can be, the carbohydrates that can be formed. One with an aldehyde group, yes, and the other one with a keto group, right? So according to the definition of the carbohydrate, I have already told you carbohydrates are aldehyde or keto derivatives of polyhydroxy alcohol. So obviously, whenever we are talking about this monosaccharides, so depending upon the number of carbon atoms, we can now classify them as aldoses and ketoses. Now for the three carbon atoms, guys, for the three carbon atoms, you already know the answer, guys. For the three carbon atoms, yes, the three carbon atoms of over here, you can see, guys, that this one, yes, for the three carbon atoms. So the carbohydrate that is having an aldehyde group, it is glyceraldehyde. Yes, it is glyceraldehyde. We have just discussed right now. Yes, we have glyceraldehyde. And the one with which is having this keto group, guys, the one which is having this keto group, you can also see this keto group over here, right? At the second carbon, keto group is always present at the second carbon. What is the name? It is dihydroxyacetone. It is dihydroxyacetone. So I hope it is clear now why we have drawn that part. Yes, just to make it clear, it is dihydroxyacetone. Dihydroxyacetone. So glyceraldehyde is having how many carbons, right? 3. Dihydroxyacetone is also having 3 carbons. But glyceraldehyde, it is having an aldehyde group present at first carbon. Dihydroxy group, it is having 3 carbons, but it is having keto group present at second carbon. So these two are nothing but these two are the carbohydrates that are formed from polyhydroxy alcohol, that is glycerol. So that is the one which is carrying 3 carbons. Now coming on to the fourth one, again, guys, we have to increase 1 carbon. The one which we have drawn for 3 carbons, like the same we will draw for the 4 carbons also. You can just practice it also. But over here, the aldoses part, yes, the tetrosis. Remember, the aldehyde carbohydrate which will be formed over here, it is erythrose. Just a moment. Yeah, it will be erythrose, right? Erythrose over here, it is the aldose wala part, right? It is aldose, right? Aldose. Erythrose, and the one which is the keto derivative of this, this, just a moment, guys. That is erythrulose. Erythrulose, right? Just remember the names erythrulose. It's a keto derivative, right? Aldehyde derivative, right? Erythrose. Erythrulose, it is keto derivative. Now coming on to the pentoses, guys, we have the 3 aldehyde derivatives. I mean the 3 carbohydrates which will be formed over here, guys. Yes, we have the ribose. We have the ribose, xylose, and yes, arabinose. Ribose, xylose, and arabinose, right? So yes, both are having this like 5 carbon atoms. Yes. And the ketosis one, it will be ribulose. Ribulose. Yes, ribulose. And xylose, it will be xylulose. We don't have any keto derivative for this arabinose, right? So we have ribose, xylose, and arabinose. Correspondingly, we will have ribulose, xylulose. Yes. Now hexoses, guys, you already know. Now these ribose, xylose, and arabinose, guys, again, they have the difference in the orientation of this H and OH. That's why at certain carbons, right? So that is why they are like the 3 forms that can be created over here. It is the ribose, xylose, and arabinose. And for hexoses, guys, you I think already know this. This is the simplest one for the day. Yes, we have the glucose. That is an aldehyde derivative. Yes, we have galactose. Galactose. We have this mannose. And yes, these are all aldohexoses, right? These are all aldohexoses, right? And on the other hand, yes, these all are aldohexoses. And on the other hand, guys, remember we have only fructose as the keto derivative. Fructose as the keto derivative, right? So it is a ketohexose. So you should remember whenever the question says the ketohexose, hexose means 6 carbons having the keto group, that is C double bond O, that is fructose only. Aldohexoses are glucose, the one which is having the aldehyde group, right? The galactose, mannose, right? All of the three will be having the aldehyde group, that is CHO. That's why they are named as aldohexoses. Hexoses means 6 carbons. Aldo means aldehydes. Aldo means aldehydes. So the first classification we have already done over here. According to the notes, it is given carbohydrates. And yes, the monosaccharides we have already discussed, guys. The functional groups aldoses and ketosis. Aldoses C double bond OH, that is CHO group, and ketosis it is C double bond O. So we have triose, tetrose, pentose, hexose, and heptosis also, like 7 carbons also, right? So we have in detail discussed about this monosaccharide. I'll be coming on to monosaccharides. Okay. Now, again, guys, the disaccharide means containing two potential sugar units. The two potential sugar units, you already know the answer. Yes, it is maltose, lactose, and sucrose. So two potential sugar units, it is maltose, yes, lactose and sucrose. Maltose, lactose, and sucrose. Maltose, lactose, and sucrose. Now, just to indicate, guys, this maltose, the two potential sugar units, they are formed from. Okay, this maltose, it is formed from what two sugar units, guys? You already know it is glucose plus glucose. Glucose plus glucose. Obviously, when the two mono sugar units will be joining together, it will be resulting in the formation of a disaccharide, guys, right? So maltose, it is glucose plus glucose. Lactose, it is galactose plus glucose. Galactose plus glucose. Yes, galactose plus glucose. And sucrose, it is glucose plus fructose. Glucose plus fructose, right? Glucose plus fructose. So that is something you have to remember over here. Out of this, one or two important questions that can be asked over here. So the reducing disaccharide, it is maltose over here. Reducing disaccharide, right? Even the lactose is a reducing disaccharide. Now, when I say reducing and non-reducing, guys, when I say what is the difference between reducing and non-reducing? Now, guys, reducing means obviously there has to be at least one free functional group. One free functional group, right? Either it can be aldehyde group. Okay, either it can be aldehyde group or keto group. Okay, either it can be aldehyde group or keto group. Okay, this free functional group is the one which makes it reducing. Now, this free functional group, it is not participating in the bond. So we have two glucose over here. We have two aldehyde groups, right? In the first glucose also, we have aldehyde group at first carbon. In the second glucose also, we have aldehyde group at first carbon. So the first aldehyde no group which is present at the first carbon, guys, it is participating, but the second glucose aldehyde group is free. It is not participating. So the first aldehyde group of this glucose, it is joining with the fourth carbon atom of the another glucose. So that is how the beta linkage is formed. But just remember over here, the second glucose over here, I mean the structure of the maltose, guys, one free aldehyde group we always have from the second glucose, and that is something which makes it reducing. Now coming on to the sucrose, guys, sucrose, it is a non-reducing sugar. Again, an important question that you have to remember, it is a non-reducing sugar, guys. It is a non-reducing sugar, means it does not have any free functional group. No free functional group. No free functional group, guys. No free functional group. So that is something you have to remember. So that is the maltose, lactose, and sucrose. You have to remember the reducing disaccharides and the non-reducing disaccharides, right? So yes. So we have discussed the disaccharides, maltose, lactose, sucrose. We have a trisaccharide also, that is raffinose, and the tetrasaccharide, right? The one containing three potential sugar units, it is raffinose. The four potential sugar units, stechoids, right? Stechoids. Okay. Now coming on to this polysaccharides, guys. Now this polysaccharides, I will be discussing it in detail. So as of now, just remember we have the homopolysaccharide and heteropolysaccharides. So these are just the classification, guys, over here that you have to remember. And I will be discussing these polysaccharides in a couple of minutes. But just if I just try to like differentiate over here these polysaccharides. Okay, let us just try to differentiate according to the definition over here. The polysaccharides, guys. Now these polysaccharides, it is containing more than 10 sugar units. That is something you already know. More than 10 sugar units. So these are classified as, yes, the homopolysaccharide, and we have heteropolysaccharide. Heteropolysaccharide, right? So we have homopolysaccharide and heteropolysaccharide. So the homopolysaccharide, it is also called as homoglycan. Homoglycan. So what does it mean? It means that it is containing one type of sugar, or I can simply say monomer units. Monomer units. So it is containing one type of sugar or monomer units. Coming on to heteropolysaccharide, hetero means different, right? Or I can simply say heteroglycan. It is also called as heteroglycan. And remember, it is containing more than one type of monomer units. Monomer units, more than one type of monomer units. So remember, the examples are glycosaminoglycans. That is, I will be discussing it in detail, guys, today. Glycosaminoglycans, right? The GAGs, the GAGs, right? Apart from that, we also have agarose, right? Which is not mentioned over here. You can mention in your notes. Agarose and pectin also are heteropolysaccharides. So this is the basic classification, guys, that we have discussed with respect to carbohydrates, right? The disaccharides, the oligosaccharides, and the polysaccharides. And yes, the homoglycans or the homopolysaccharides have starch, dextrin, glycogen, cellulose, inulin. Heteropolysaccharides containing more than one type of monomer units, we have hyaluronic acid, heparin, chondroitin sulfate, dermatin sulfate, and keratin sulfate. Right? So we are going to discuss it one by one. So let's just, yeah, the first question is done over here, right? So I hope you all understood. Coming on to the next one. Now, what are the different types of the monosaccharides and their biological importance, guys? We have already discussed the different types of monosaccharides. Now, I have already told you, based on the number of the carbon molecules, we can have 3, 4, 5, 6, 7, 8, and even 9 also. We have trioses, right? Tetroses, pentoses, hexoses, like this we have already discussed. Now, what is meant by this monosaccharide, guys? What is actually the definition of this monosaccharide? So even after no they cannot be further reduced, guys, even after no adding water or something. They are called as simple sugars. Right? Now, what is the general formula, guys? The general formula over here, remember, the general formula, it is CN, yeah, H2ON. We have already told you it's hydrates of carbon. Hydrates mean water, and that is the carbon. N represents the number of the carbon molecules, the number of carbon atoms, right? The number of carbon atoms over here. So that is the general formula for this monosaccharides, right? So yeah, I have already told you the very important characteristic over here that they cannot be further hydrolyzed. They cannot be further hydrolyzed, right? So monosaccharides are categorized based on the functional group and the number of carbon atoms. We have already discussed this. Yes, based on the number of carbon atoms, I have already told you guys right now. Yes, based on the number of the carbon atoms, yes, we can classify it as triosis, tetrosis, pentosis, and hexosis. And depending upon the functional group, these are aldehyde or ketosis. So we have the three carbon atoms, the aldehyde group, the carbohydrate will be glyceraldehyde. The three carbon atoms having keto group, it will be dihydroxyacetone. And how they are formed, we have already seen. Again, the 4-1, erythrose, erythrulose, pentosis, ribose, xylose, arabinose. On the other hand, the keto derivative of this, it will be ribulose, xylulose. Hexosis, we have glucose, galactose, and mannose. And on the keto derivative, yes, we have fructose. That is a keto hexose, right? The keto group, it is present at the second carbon. The aldehyde group, it is present at the first carbon. So that is what is written over here. Yes, they are monosaccharides with aldehyde functional group with the keto functional group. Aldehyde, we have already discussed the glyceraldehyde. That is the one which is containing how many carbon atoms, guys? It is containing 3 carbon atoms, right? And glucose, it is containing 6 carbon atoms, 6 carbon atoms. And glucose is an aldohexose or ketohexose? Yes, it is a aldohexose. Even yeah, it is an aldohexose. Aldo means aldehyde derivative containing 6 carbon atoms. Glyceraldehyde, 3 carbon atom. That is the parent carbohydrate. Ketosis, we have already discussed dihydroxyacetone. Yes, I have already told you it is having 3 carbon atoms with the keto group present at second carbon. And fructose, yes, it is an yes, keto hexose. Keto hexose, why? Because it is containing 6 carbon atom, that is hexose, with a keto group at second carbon. That's the structure of fructose, right? So they are further classified as triosis, tetrosis, pentosis, hexosis, heptosis based on the number of carbon atoms, right? We have already discussed these 3 points. Now, these classifications, along with the functional groups, are in the naming of the monosaccharides. So remember, guys, we have already told you this is very important that glucose is aldo. Aldo means aldehyde group. Hexose means 6 carbon atoms. So yeah, so we are using the carbon atom and the functional group. So both are used together to give the name, right? And fructose is a keto hexose. Keto means C double bond O present at second carbon only. We have already seen. And hexose means 6 carbons, right? So that is the basic the important points regarding with respect to the monosaccharides. Now coming on to the important concepts over here, guys. What is so important over here that is given in the notes? Triosis means, guys, you already know we have how many carbon atoms? Yes, we have the 3 carbon atoms, right? Now these 3 carbon atoms, if they have an aldehyde group, right? If they have an aldehyde group, it is glyceraldehyde. If they have a keto group, it is dihydroxyacetone, right? Now, this both are found in cells as phosphate. They are both found in cells as phosphate. And remember, guys, this glyceraldehyde 3-phosphate. Okay, they are not found alone. They are found with linkage to this phosphate, right? So glyceraldehyde combined with phosphate will be glyceraldehyde 3-phosphate. It is an intermediate product in the glycolysis. Glycolysis, guys, we have already discussed in detail earlier also. And you already know in glycolysis, you already know from glucose what we are forming. Yes, we are forming pyruvate, right? So that is something called as the aerobic glycolysis. And in this aerobic glycolysis, an intermediate product that you will encounter over here, that is glyceraldehyde 3-phosphate, right? Now coming on to tetrosis, guys. Yes, we have erythrose. Erythrose, I have already told you, is an aldose sugar, right? The aldose sugar because it is containing aldehyde group. We have how many carbons now? 4 carbons, and it is having aldehyde group at first carbon. First carbons. So it is present everywhere, right? It is occurring everywhere in the body. Now it's it's an intermediate in the carbohydrate metabolism only. We'll just be um, seeing this in the later parts of the metabolism, right? Apart from that, guys, ribose, Ribose, like the pentoses, right? We have how many carbons? 5 carbons, right? And the one which is having aldehyde group at first carbon, it will be ribose, right? So it's an aldehyde derivative. Now, ribose from R, remember it is constituent of ribonucleic acid, right? And nucleotides. Nucleotides are again made up of phosphate, sugar, and base, right? So remember, guys, for the structure of the RNA and nucleotide coenzymes like ATP, NAD and NADP, right? And even for the structure of this, yeah. So that is like you have to remember these are an important part of the structure of this RNA, guys, right? Deoxyribose DNA, guys, no need to tell. They are responsible for the formation of the structure of the DNA, right? Ribulose. Now, when I say ribulose, guys, again, you have to remember ribulose is also having 5 carbons. Now, what is the difference between ribose and ribulose, guys? So ribulose will be having the keto group at second carbon. So we have the difference in the functional group. Ribulose will be having, it's a keto derivative, guys. It's a keto derivative. Now, when I say ribose, guys, it's an aldehyde derivative. It is an aldehyde derivative, right? So that is the difference between this ribose and ribulose, guys, right? So that is something you have to remember, right? So this ribulose, it is produced during metabolism and will be, yeah, no, we are gonna discuss one of the important shunts, that is the hexose monophosphate shunt, and that is you will see this particular ribulose which will be formed as a intermediate product over there. Now we have xylose, right? Xylose, basically it is having 5 carbons only, right? And it's having this aldehyde group, right? And it's a constituent of glycoproteins. Now, when I say glycoproteins, guys, you already know I'm talking about glycogen plus proteins, right? Glycogen plus proteins and gums, right? So remember, it is involved in the function of the glycoproteins. We have xylulose. Xylulose, it's a keto derivative. Xylulose is an intermediate in the uronic acid pathway. It's an very important. It's an intermediate in the uronic acid pathway, right? So it is excreted in urine, and it is, yes, then essential pentosouria, right? And that is something you have to remember. Lyxose, it is found in the heart muscle, guys. Again, an important part, and it's a constituent of lyxoflavin of the heart muscle, right? So that is about the ones which we have discussed, the triose, tetros, and pentose. Coming on to the hexoses, guys, the most important ones that you have to remember, the glucose. Glucose, I even not need to tell you, they are a constituent of polysaccharides. Now, when I say poly means many sugar units. So it is a constituent of starch. Starch is formed of glucose. Even the glycogen, it is formed of glucose. Cellulose, it is formed of glucose. So this glucose is helping in the formation of the starch, glycogen, cellulose, and even the disaccharides, guys, we have already discussed, right? The maltose, the lactose, and sucrose, right? They all are having glucose as their one of the units, right? Even in the fruits also, right? So it is the fuel of the life, right? So yes, remember, it is the structural unit of cellulose in plants, right? D-galactose. And remember, guys, in diabetes mellitus, also this glucose, there is excess glucose in the body, so it is excreted via urine. So yes, galactose, guys, you already know it's a constituent of lactose, that is the milk sugar, right? We have already seen this disaccharide lactose, that is galactose plus glucose, and that lactose is nothing but the milk sugar. So it is converted to glucose, and yes, if it is, you might have encountered certain patients who are no coming up to you with complaints of intolerance to lactose. Intolerance to lactose, guys. So that is something that you have to remember, that in the bloods, the galactose levels will be raised. Galactosemia, it will not be. And next is mannose, right? So it is found in plant polysaccharides and animal glycoproteins. And fructose, yes, it's a constituent of, right, sucrose and inulin, right? So remember, yeah, the fructose, it's an intermediate of glycolysis only from glucose. Yes, what is formed? Yes, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6 biphosphate. So this fructose, it is basically in the glycolysis pathway also, right? Now, Pseudoheptulose, guys, it is found in the plants. So it is an intermediate product in the hexose monophosphate shunt, right? We have already discussed the monophosphate shunt earlier. So that is a basic about this monosaccharides and their importance, guys. Most of them, they are like the intermediate products in the metabolic pathways running in our body, right? And apart from that, they are an important constituents of various molecules, right? And that is something that you have to remember for the exam. So this flow chart or this table is like essential. This I have taken from the notes, and like you can just copy paste it in your exam. So you just have to learn, revise the concept, and that will be easier. Okay. Now coming on to the different aneurysms for carbohydrate. Okay. Before mhm, okay, coming on to the isomerism part, let us just discuss the polysaccharides, guys. Let us just try to focus on the polysaccharides right now, and after that I will be like discussing this that part. So we the most important part over here, it is the homopolysaccharides, guys. The homopolysaccharides, right? So, and the heteropolysaccharides. So let us just try to discuss something about the homopolysaccharides also and the heteropolysaccharides also. Now, when I say homopolysaccharides, guys, you already know it is also called as homoglycan. It is also called as the homoglycans, right? It is containing one type of sugar units, one type of sugar or the monomer units, right? Monomer units. So the example, you already know the homopolysaccharides. It is like the starch, the glycogen, the dextrin, cellulose, inulin, dextrin, and chitin. Now, coming on to the very first one, guys, that is the starch, guys. Now, when I say starch, guys, I'm discussing the homopolysaccharides right now. Then I will be coming on to the heteropolysaccharides. Now, when I say starch, guys, so starch is nothing but the storage form. Remember, it is the storage form of carbohydrates in plants, right? You already know it is the storage form of carbohydrates in plants. So this starch, it is divided into amylose and amylopectin. Now, what is the difference? This starch, okay, let us just try to tabulate the difference over here in a very short manner. Yes, we have amylose and we have amylopectin. Amylopectin. Now remember, guys, these are mostly the branched molecules, right? So remember that over here in amylose, guys, remember it is soluble, but it is unbranched, guys. It is soluble, but it is unbranched. It is soluble, but it is unbranched, guys. Right? So it is just like in this form, guys. So the sugar molecules are like, they are added like this. Let me add it with another color, guys. So you will be like more, yes, the sugar molecules are like this. They are soluble, but they are unbranched, right? So these are nothing but the glucose molecules, the glucose molecules. Now, apart from that, if we talk about amylopectin, guys, amylopectin, it is insoluble. It is insoluble. Plus, it is highly branched. Plus, it is highly branched. So you can very well see that, yes, we have these glucose molecules. And yes, we have several branching presents at different levels. So that is the structure of amylose and amylopectin, right? Amylose being the unbranched one, and the branched one being the amylopectin. Amylose is soluble, amylopectin is insoluble. Just one words if you just want to remember about starch, guys, right? The storage form of carbohydrates in plant, and sir has told about two starch molecules, amylose and amylopectin. Amylose, which is soluble, unbranched, amylopectin, which is insoluble, and it is highly branched, right? Now coming on to the glycogen wala part, guys. Now, when I say glycogen, guys, when I say glycogen, you can very well see over here, this glycogen, guys, it is made up of alpha D glucose. Remember, guys, it is made up of alpha D glucose. Alpha D glucose, right? I'll be coming to this D and L glucose. So just remember over here, it is made up of alpha D glucose. And secondly, remember it is a branched polymer. It is a branched polymer, branched polymer. And one more important thing that you have to remember over here, that is the storage form of carbohydrates in animals. Storage form of carbohydrates in animals, guys, that you already know. It is also called as the animal starch, right? It is also called as the animal starch. It is also called as animal starch. The two important viva questions that can be asked over here. The two important viva questions that can be asked over here. The glycogen, it is stored in. Glycogen is stored in two sites in our body. Now, the two sites, again, very, very important. The first one being the liver, the other one being the muscle, liver and the muscle. So in the liver, we have the aggregates of beta particle. We have aggregates of beta particles, right? So that is the rosette of the beta particles, right? In muscle, basically it's in the form of the beta particles, the first one being the rosette, and over here in the form of beta particles. And yes, beta particles, remember the single beta particle. Again, an important question. The single beta particle has 60,000 glucose units. Glucose units. So remember, if I simply ask you the total content of glycogen, it is more in, again, an important question. The total content of glycogen, it is more in muscle. But if I talk about percentage by tissue weight, glycogen is more in liver. Glycogen is more in liver because the rosette of the beta particles, they are highly condensed or they're compactly packed, guys. So we have two different questions. I want you to mention in your notes, guys. These are two important questions. So the total content of glycogen, obviously we have more muscle in the body. So the total content of glycogen, it is more in muscle. It is more in muscle. But when I say liver, right? So the percentage by tissue weight, if I just compare with like percentage by tissue weight, remember glycogen is more in liver. Glycogen is more in liver. More in liver, right? So remember the rosette of the beta particles, right? They are highly condensed. They are highly condensed, guys. They are highly condensed. So that is about a little information about the glycogen. That is another, yeah, the another homopolysaccharides. Now coming on to the cellulose, guys, right? You already know the cellulose. It is the major dietary fiber, right? The major dietary fiber. Two, three things that you have to remember. Major dietary fibers. Yes. And remember over here, it is composed of beta D glucose. Beta D glucose, right? Just small things that you have to like mention over here. The beta D glucose and yes, over here we have one linkage that is called as beta 1 to 4 linkage. Beta 1 to 4 linkage. And remember one important viva question that you have to remember from exam point of view, that the human lacks enzyme to hydrolyze this beta 1 to 4 linkage. That is why we are not able to digest cellulose. And this enzyme, it is nothing but the cellulase. It is nothing but the cellulase. So we lack this enzyme. That's why we are not able to digest these plants, right? So that is again very, very important. So these are the important three that we have discussed: the starch, glycogen, and the cellulose, right? In a very short form. And that are all the important MCQs also that you can remember. And even in the short answers, you can just mention. Apart from that, guys, if you just want to remember about yeah, the chitin, guys. So the chitin, it is so chitin, it is the homopolysaccharide, right? We have already discussed the homopolysaccharide. So this, it is found in exoskeleton of insects, right? It's found in the outer skeleton of the insects. And yes, so remember, it is formed of N-acetyl glucosamine. So that is something you have to remember about chitin. Okay. Now remember, guys, dextrin, remember it is the hydrolytic product of starch. hydrolytic product of starch, guys. And remember, this dextrin is nothing but oligosaccharide. So yes, we have just classified over here, but remember it's an oligosaccharide, guys, right? So yes, so that is something that you have to remember. Even the inulin, guys. So inulin beta, remember. Okay, one important point that I can tell you over here, inulin, it is a polymer of beta D fructose. Beta D fructose. Actually, it is fructosin. Now remember, guys, it is used as an external agent to check the GFR, that is the glomerular filtration rate, right? So that test, it is called as the inulin clearance test. So this inulin, it is no exclusively abundantly found in dahlia, chicory roots, even the garlics, they will have this inulin much more. So it's used as an external agent. So it is used as an external agent to check for GFR, right? The glomerular filtration rate, and test. Yes, the test, it is called as the inulin clearance test. Inulin clearance test. Inulin clearance test, guys, right? So inulin, it is majorly, it is no found in dahlia, chicory roots, and even in the garlic, guys, you will see this inulin, right? So that's found in dahlia, chicory roots, and garlic. So that is about the homopolysaccharides. That is about the homopolysaccharides that we have discussed. Now coming on to the heteropolysaccharides and its features. The heteropolysaccharides and its feature. Now, now. Let us just try to simplify this information that is given over here in a very short manner. Like let us just try to understand what does this whole piece of information says. So yes, we have the heteropolysaccharides. Heteropolysaccharides. Now this heteropolysaccharides, it is also called as, right? It is also called as heteroglycans. We have already discussed it is also called as heteroglycans. Remember, it is made up of more than one type of monomer units. More than one type of monomer units. It is made up of more than one type of monomer units, right? So these are nothing but the amino sugars and the uronic acids, right? Now, these are commonly known as glycosa glycosaminoglycans. That is commonly called as GAGs, right? Now, these heteropolysaccharides, they are also called as, yes, they are also called as mucopolysaccharides. Mucopolysaccharides, right? Now, why they are called as mucopolysaccharides? Now, when I say muco, guys, muco means mucus, right? So they are present in the mucus secretions, guys. That is again, mucus is for lubrication, right? The mucus, that is for lubrication, guys. So these heteropolysaccharides, they are also called as mucopolysaccharides because they are present in the mucus. No, all GAGs, the glycosaminoglycans, they combine with proteins to form proteoglycans, right? Proteo means protein, glycans means this, glycosaminoglycans, right? They are highly sulfated. They contain more negative charge. They are slimy and slippery. So these mucopolysaccharides or glycosaminoglycans are heteropolysaccharides containing uronic acid and amino sugars, right? I told you it's more than one type of monomer units, right? The sugar derivatives, these are the amino sugars or the uranic acids, right? Apart from that, we have the acetylated amino groups, sulfate and carboxyl groups that are also present, right? They attract the water molecules, guys, so they produce viscous solutions. Now, apart from that, you should remember the mucopolysaccharides. the one secreting the mucus, they combine with protein to form the mucoproteins. They combine with protein to form the mucoproteins. Right? So that is something you have to remember over here. Now, coming on to the examples of this mucopolysaccharides or heteropolysaccharides or glycosaminoglycans, the very first one that you have to remember, it is the glycogen. It is the glycogen, guys. Just a moment. Yes. Sorry, glycogen, I'm saying hyaluronic acid. Yes, the hyaluronic acid, right? Now, hyaluronic acid, guys, remember the location, guys. Again, a very important Viva question. It is present in the connective tissue, the tendons, guys, the synovial fluid, and the vitreous humor in the eyeball, right? So they are present in connective tissues, tendons, right? The tendons which helps in the connection of the muscle to the skeleton, right? The muscle to the bone. The tendons, the synovial fluid, guys, right? In the knee joint, in the this elbow joint, right? And in the vitreous humor of the eye, we have the aqueous humor and the vitreous humor of the eyeball. So in the vitreous humor, we have this hyaluronic acid. Now it acts as a lubricant in the joint cavities. That's why it's present in the synovial fluid and other parts, right? Apart from that, guys, it is formed of repeating units of N-acetyl glucosamine, right? And glucuronic acid, right? Yeah, the amine sugar, that is the N-acetyl glucosamine, and the uronic acid, that is the glucuronic acid, right? Apart from that, heparin, I know everyone knows about the heparin, guys. Yes, it is an anticoagulant. So it is widely used in taking blood in vitro for clinical studies. Like we are taking blood, we don't want the blood to clot, so we'll add heparin, guys. We'll add heparin in the test tube earlier, right? So that's an anticoagulant. It is used in vivo to prevent intravascular coagulation. Obviously, intra means inside, vascular means the blood vessel. So inside the blood vessels, right? So yes, inside the blood vessels. So it is also used in vivo to prevent the intravascular coagulation. Now remember, guys, it activates anti-thrombin 3, which in turn activates thrombin, right? Factor 10 and factor 9 in the coagulation pathway, guys, remember? So heparin, it is present in liver, lungs, spleen, and the monocytes. The WBCs, that is the monocytes. Chondroitin sulfate, guys, remember it is the ground substance of connective tissue. We know that the connective tissue is made up of cells and the ground substance. So the ground substance, it is nothing but the chondroitin sulfate. So chondro basically means cartilage, guys. So they are present in cartilage. That's the chondrocytes. Yeah, cartilage chondrocytes, the cells of the cartilage chondrocytes. Chondroitin sulfate, it is present in cartilage, the bones, the tendons, the cornea. So remember chondroitin, cornea, and skin. Skin. Keratin sulfate, I think you remember, it is the only glycosaminoglycan which does not contain any uronic acid. Again, a very important question. So they will simply ask you a glycosaminoglycans which does not contain any uronic acid. It is keratin sulfate, right? So the repeating units are galactose and N-acetylglucosamine in beta linkage. And remember the keratin sulfate, it is found in cornea and tendons. Dermatin, derma means the skin, guys. It is found in skin, the blood vessels, and the heart valves, right? And it is helpful in maintaining the shape and structure. Valves of the heart are one which is preventing the backflow of the blood, right? The bicuspid valve, the tricuspid valve, right? So remember, that is the dermatin sulfate, which is found over there as well. So that was regarding this heteropolysaccharides. Now coming on to the one which I left, guys. Yes, the types of the structural isomerism. The types of the structural isomerism. Now, before starting with that, let us just try to know. Let us simplify, guys. What does this isomerism in carbohydrates mean? Let us just simply discuss in a very short term, and then we'll discuss to the question as well. Isomerism in carbohydrates. Now, when I say isomerism, So it is basically of two types. Yes, we have structural isomerism. Yes, we have structural isomerism, or it is also called as stereoisomerism. And the other one, we have the optical isomerism, right? So we have structural isomerism and optical isomerism. Now, what is the difference, guys? Now, when I say structural isomerism, remember I'm talking about it has the same molecular formula, and different structural formula, different structural formula. And when I say optical isomerism, guys, it is difference in rotation of plane polarized light. So let us just try to discuss one by one. Now, any if I say the carbon atom, guys, okay. In a structure of the glucose, guys, you already know we have 6 carbon atoms, right? We have 6 carbon atoms in the structure of glucose, right? First carbon, second carbon, third, fourth, fifth, sixth. Now, when I say asymmetric carbon atom, guys, when I say asymmetric carbon atom, so I am telling that all the four valencies, basically it is surrounded by 4 different kind of groups. 4 different kind of groups, and that is something called as an asymmetric carbon atom. Now, if I talk about the carbon atom 2, guys, over here at the first place, it is like this. The second place, we have this OH, H over here, right? Third place, OH is on this side, right? So you can see this second carbon atom, yeah, it is surrounded by 4 different valencies. The first part is this, the second is this. So we have 2 different groups. The third is this, and the fourth one is this. So second carbon atom, it is an asymmetric carbon atom, which is surrounded by like 4 different kinds of groups, right? So remember that is something you have to remember, right? Now the structural isomerisms can be divided into the 3 types, guys. The structural isomerisms can be divided into 3 types. And in short, you should remember we have the D and L isomerism. D and L isomerism, right? We have enomerism, the second one, enomerism. And the third one, that is the epimers. Now, when I say DL and L isomerism, let us just try to discuss in very short about the D and the L isomerism. Now, D and L isomerism. Now, if I just want to discuss the simplest carbohydrate, now when I say simplest carbohydrate, yes, you already know it is glyceraldehyde. Glyceraldehyde. It is having 3 carbon atoms, right? It is considered as the parent carbohydrate. So you already know we have this, the 3 carbon atoms, right? Yes. Now, just try to see what I'm telling you, DL and L isomerism and the parent carbohydrate. So we have 3 carbon, first, second, and third. So that basic structure that we have drawn, it is called as the D-glyceraldehyde, right? D-glyceraldehyde. So remember guys, over here we can find only 1 carbon atom, that is penultimate, right? Right? 1 carbon atom, that is penultimate, that is the second carbon atom, guys, right? So remember the number of the structural isomers, guys, you already know the number of the structural isomers. Okay, I hope you already know the Liber-Von-Hoff rule, right? So the number of structural isomerism, that is 2 raised to the power n, and n represents the number of asymmetric carbon atom. The number of asymmetric carbon atom, and you already know the number of asymmetric carbon atom over here will be the second carbon only. So we have 2 raised to the power 1, we have only 2 structural isomerism. That is the penultimate carbon atom, that is the C1. So we have the D-glyceraldehyde, and similarly we will have this L-glyceraldehyde. This OH will shift to this part. Yes, and then it is just the same. So it is L-glyceraldehyde, right? Now, if we talk about, yeah, the structural isomers of no glucose. Now, glucose, if it is in the ring form, guys, if it is in the ring form, remember it has 5 asymmetrical carbon atoms. 5 asymmetrical carbon atoms, and yes. So if it is 5, so the number of structural isomers, guys, what it will be? Structural isomers 2 raised to power n, 2 raised to power 5, it will be 32. Now, 32 structural isomers of glucose are possible, guys. Now, again, if in the question 32 is not given, guys, now considering glucose as straight chain, remember in the straight chain, we have only 4 asymmetrical carbon atoms. 4 asymmetrical carbon atoms, and over here 2 raised to power like 4, that will be 16, guys, right? So it is C2, C3, C4, C5, right? In ring form, C1 also acts as a asymmetrical carbon atom, but in straight chain it is not. In straight chain it is not. C1, it is not, like in straight chain it is not an asymmetrical carbon atom, because 99% of the glucose in solution exists in the ring form. So because if in our body, if we are considering only glucose, guys, our body is predominantly made up of water, okay, which means the form of glucose which is present in our bodies, it is in solution form. So 99% it is existing in the ring form only, and obviously ring form means how many asymmetrical carbon atoms? 5. So the answer will be 32. If it is not given in the option 32, go with 16, considering glucose as the straight chain. We have only 4 symmetrical asymmetrical carbon atoms. So that was about the D and L isomerism, right? Now, coming on to one of the parts that we have discussed over here, that is the, yes, yes. One important part that I have to discuss over here, that is the enomerism, guys. That is the enomerism, right? So we have already discussed, according to our part, we have discussed the D and L isomerism. Now coming on to enomerism, guys, let us just try to discuss one of the question over here, enomerism. Now, what is this enomerism? Enomerism. Now, what does this mean? It means that there is isomer or isomerism at functional carbon atom. At functional carbon atom, right? So remember in aldoses at C1 carbon atom, and in ketosis at C2 carbon atoms. Now, if you know the basic that we have discussed in the previous classes, guys, in the live sessions on the YouTube which I conducted, I have told you about the ring structures. No, no. So let us just try to draw a very simple ring structure over here, right? So this structure, it is of glucopyranose, right? So we are discussing examples over here. So we have alpha glucose and beta glucose. We also can have alpha fructose and beta fructose. Now we are drawing 2 glucopyranose. Now at the C1, guys, at C1, remember if the OH group, it is present above, Okay, so that structure it is called as beta glucopyranose. Remember, guys, birds fly in the sky high, right? So birds fly in sky. Now birds means beta, sky is above only. It means the OH it is above, right? So above it is beta glucopyranose. Now if at the C1, the OH it is down, guys. If the OH is down, it is, yes, the alpha glucopyranose. So we are pyranose. We are just discussing the ring, guys. We are just discussing the ring over here, right? The glucose ring, that is the pyranose ring, guys. So it is glucopyranose ring, beta glucopyranose ring, and alpha glucopyranose. It's the structure, the glucose and the ring form the beta and the alpha. And how can you differentiate? That is the functional carbon atom, which is like making the difference, right? Now, one property of merism that you have to remember, guys, that is something called as muta rotation. Muta rotation, guys. So it is actually rotation, change in the rotation of plane polarized light. Plane polarized light. So remember, guys, the alpha glucose, it is plus 112 degree, beta glucose plus 19 degrees, right? So this is because of alpha to beta interconversion. So that is yeah, that you have to remember, guys. So yeah, that is something you have to remember. Okay, so that is one of the properties of this, that is muta rotation, right? No, that is about the anomerism. So the linear structures to the cyclic structures we have already discussed, right? So remember the pyranose ring, the six-membered ring, and the number of carbon atoms, they are five, right? The number of carbon atoms, if you just see over here, the C2, C3, C4, C5. The number of carbon atoms, it is five. Furanose ring, basically I've told you the fructose wala ring, five-membered ring, and the number of carbon atoms, it is four, 1, 2, 3, and 4, right? So that is something you have to remember about the anomerism, guys, right? Now coming on to the third one, that is the epimers, guys. That is the epimers, right? Or epimerism, guys. Epimerism, right? So let us just try to discuss the epimerism also. That will like complete our topic for today. That is the epimerism, or remember, guys, it is isomerism at a single carbon atom. Isomerism at a single carbon atom, right? Other than functional and penultimate carbon atom, right? The one with the asymmetric, right? Now let us just try to understand this with the help of a structure. You already know the structure of glucose, guys, the basic structure of glucose. So we have 1, 2, 3, 4, 5, 6. At the first place, we have the aldehyde group, right? Second place, OH is over here. This is H, this is OH, OH, OH. At the third place, only the OH is directed to the other side, otherwise the structure remains the same, right? So that is the basic structure of the glucose, right? Now from the glucose, guys, if I'm changing the orientation only at the second carbon atom, right? If second carbon atom is involved, if second carbon atom it is involved. Now the first, it will like be the same. The second one, that is H, yes, okay. OH will come to this side, and the other structures will remain the same. The other structures will remain the same, right? So that is OH, 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 H2, H, and H. So what we have changed is the second wala carbon. That's the second wala carbon. The other is same. That this structure it is called as mannose. So yeah, the mannose I can simply say it is C2 epimer of glucose. It is C2 epimer, carbon 2 epimer of glucose, right? It is C2 epimer of glucose. Now, if let us just try to see one more change over here, guys. Let us take one more example over here, right? If the change is at the fourth carbon atom, now again, the first carbon atom will be same. The second from the glucose, it will be like same. The third also, it will be OH will be on the, yes, the left hand side, right? The fourth carbon atom, OH is on the right. So yeah, obviously it will go towards the left, right? Whereas the other structures will remain the same, right? So this structure, it is called as galactose. Galactose. Remember, it is the fourth epimer of glucose, carbon fourth epimer of glucose, guys. Glucose, right? So only C2 and C4, right? Mannose and galactose. So these, yeah, remember, guys, the mannose and galactose. So remember, these are examples of diastereoisomers, right? So it is not including the penultimate or more than one carbon atom. So obviously they are not mirror images. They are not mirror images, right? If guys, they are having a difference in existing in all the three carbon atoms, C2, C3, C4, like, or C5 also. So together, like, then it's a mirror image. Then it is called as an enantiomer. But over here, it's like the same one. So remember, that is about the epimerism, guys. Let us just try to read it from the notes also, what is given over here. Yes, the epimerism, it is always exogenous, right? Difference in the H and OH orientation we have already discussed. Mannose is an epimer of glucose at C2. Galactose is an epimer of glucose at C4. And how it is, I have already shown you, guys. I have already shown you this epimerism, right? So whatever is there in the notes we have discussed. So mannose and galactose are not epimers of each other. So that was regarding this. So today we have already discussed, guys. Let us just try to summarize the classification of the carbohydrates, the basic definition of the carbohydrates, how are they formed, the classification mono, di, oligo, polysaccharide. The monosaccharides we have discussed in detail along with the biochemical importance, right? We have discussed the isomerism part, right? We have discussed the isomerism, the structural or the stereoisomerism, the DNL isomerism, right? We have discussed enomerism, epimers, right? We have discussed the polysaccharides in detail, the homopolysaccharides and the heteropolysaccharides. So that's all for today, guys. So that's all for today. So these are the social media handles. You can just follow for the further updates of classes, right? And thank you, guys, for your patience, for listening me out and for sparing your time, right? So thank you.